Hi everyone, welcome to Bite Size Med, where we talk about quick bite-sized concepts in basic medical sciences for study and rapid review. This video is on how the kidneys regulate acid-base balance. Just a quick recap. There are three regulatory systems of acid-base balance: buffers, the lungs, and the kidneys. The Henderson-Hasselbalch equation gives a pH of the solution. That's pH equals pK plus the log of the concentration of base over acid. If there's an increase in the hydrogen ion concentration, the pH reduces, and there's acidosis. If there's a lower hydrogen ion concentration, the pH rises, and that's alkalosis. The normal pH of blood is around 7.4. The most important buffer pair is the bicarb carbon dioxide pair. They have a pK of 6.1, so the pH is equal to 6.1 plus the log of the bicarb concentration over 0.03 into the partial pressure of arterial carbon dioxide. This bicarbonate is regulated by the kidneys. The kidneys are the slowest to respond, but they're the strongest. They manage high hydrogen ion concentrations and low bicarbonate by excreting more hydrogen ions and producing new bicarbonate. Similarly, low hydrogen ions and high bicarbonate is managed by reducing hydrogen ion secretion and excreting more bicarbonate. So first, let's see how these two get exchanged in the nephron. Normally, hydrogen ions are secreted and bicarbonate is reabsorbed. So this is the nephron with the glomerulus, the proximal convoluted tubule, the loop of Henle with its thin descending limb, thin ascending limb, thick ascending limb, the distal convoluted tubule, and the collecting duct. Except for the thin descending limb and the thin ascending limb, all other parts of the tubule are involved. So the proximal convoluted tubule, the thick ascending limb, the DCT, and the collecting duct. So we're going to look at it in two parts: bicarbonate reabsorption first, and then hydrogen ion secretion. 80% of the bicarb gets reabsorbed in the early PCT. Carbon dioxide enters the cell, binds to water, and by carbonic anhydrase becomes carbonic acid. That dissociates into the hydrogen ion and the bicarb ion. The hydrogen ion gets secreted into the tubular lumen by a sodium-hydrogen exchanger. This is a secondary active transport. The energy for this comes from the sodium-potassium ATPase on the basolateral membrane, which pushes 3 sodium out and 2 potassium in. So now the sodium concentration in the cell is low, which pulls sodium along its gradient in exchange for the hydrogen ions. Bicarbonate gets reabsorbed with sodium as a cotransport, while in the later nephron, it gets exchanged for chloride. So the hydrogen ion that's secreted, it binds to bicarbonate, which has been filtered from the glomerulus and so is in the tubular lumen, and by carbonic anhydrase, they form carbonic acid. That then dissociates into carbon dioxide and water, and the carbon dioxide diffuses back into the cell. So what's happening here? For every bicarbonate that's getting reabsorbed, 1 hydrogen ion is getting secreted. The bicarbonate gets into the bloodstream, but the hydrogen ion binds to the filtered bicarbonate in the lumen. So there's no net secretion of hydrogen ions, but there is net reabsorption of bicarbonate. So that's bicarbonate reabsorption. Now, hydrogen ion secretion is more in the late DCT and collecting duct. In the type A alpha intercalated cells, carbon dioxide enters the cell, combines with water to form carbonic acid, which then dissociates into a hydrogen ion and the bicarb ion. The hydrogen ion here gets actively pushed into the tubular lumen by a hydrogen ATPase and a hydrogen potassium ATPase. This is primary active transport because it uses ATP versus the PCT, where hydrogen was secreted as a part of secondary active transport. And here the bicarbonate gets reabsorbed in exchange for chloride. When the secreted hydrogen ions is more than the bicarbonate in the filtrate, excreting it as an ion itself will lower the urinary pH. That number can go down to as low as 4.4. This is the limiting pH. It cannot go lower than that. The body produces volatile acids like this carbonic acid and carbon dioxide, but also catabolism of proteins and phospholipids produces fixed acids, which cannot dissociate, but these have to get excreted as well. But the hydrogen also needs to get out. So how? It gets buffered by two urinary buffers, the phosphate buffers and the ammonia buffers. The secretion of a hydrogen ion with the urinary buffer is called a titratable acid. The phosphate buffer involves inorganic phosphates. They are filtered by the glomerulus, so they're in the tubular lumen. They bind to the hydrogen ions to form this titratable acid. Phosphates do get reabsorbed by the proximal convoluted tubule, but because more water is reabsorbed there, the phosphate concentration in the lumen increases. Now, this bicarbonate that is generated is new. It's not just replacing what was filtered, since the hydrogen ion joined a buffer that is not bicarbonate. So we've effectively created a new bicarbonate molecule. But since phosphate does get reabsorbed, the amount here is small, and that's why there's an ammonia buffer. Amino acids get metabolized in the liver and form glutamine. In the PCT, glutamine gets taken up by diffusion down its concentration gradient. By glutaminase, it becomes glutamate, and by glutamic dehydrogenase, it forms alpha ketoglutarate. Each of these steps releases an ammonium ion, and further metabolism of this alpha ketoglutarate produces two bicarbonate ions. So in the end, what we need to know is there's two ammonium ions and there's two bicarbonate ions. The bicarb gets reabsorbed, and this is new bicarb. Ammonium shifts out of the cell by countertransport with sodium. It binds with chloride and gets excreted as ammonium chloride. The collecting ducts are permeable to ammonia. So ammonia can diffuse into the tubular lumen, bind with the hydrogen ion that's been secreted to form ammonium. But ammonium cannot go through the luminal membrane, so it gets conveniently trapped in the lumen. This is called diffusion trapping. So it binds to chloride, forms ammonium chloride, and gets eliminated. It's this ammonia buffer system that can be upregulated when needed, like in long-term chronic acidosis. So secretion of hydrogen ions and reabsorption of bicarbonate go together. So what can affect this? First, the partial pressure of carbon dioxide. If it's high, like in respiratory acidosis, by the Henderson-Hasselbalch equation, the pH will be low. So the kidney compensates by reabsorbing more bicarbonate and excreting more hydrogen ions. So it compensates for respiratory acidosis. If the PCO2 is low, like in respiratory alkalosis, the pH is high, and the kidneys compensate by reducing bicarb reabsorption and reducing hydrogen ion excretion. And that's how it compensates for respiratory alkalosis. If the blood hydrogen ion levels are high and the bicarb is low, like in metabolic acidosis, the kidney will compensate by removing the excess hydrogen ions and keeping more bicarbonate. And it does the opposite in metabolic alkalosis. So the kidneys can compensate for all four primary acid-base disturbances. And that is how the kidneys are responsible for acidification of urine and also regulate acid-base balance. If you like this video, give it a thumbs up and subscribe to my channel. Thanks for watching, and I'll see you in the next one.